It's 12:30. It is December 4th. This is our last class period before our final exam. If everyone remembers or does not remember, our final exam is going to be administered on Tuesday, the 9th, as early as 7:30 in the morning and as late as 8 p.m. You'll have 2 hours in which to take the exam. Asma, what's the best way to prepare for the final exam? The best way to prepare for the final exam is going to be to do the practice exam, which will be posted by 5 p.m. today. In addition to that, kind of in a more general, maybe more holistic way, I'm going to go ahead and get a full screen here so I can write on it. I'll do it. Sorry to delay this, but the way that I think is really helpful to prepare for and review this material is, and this goes for any class for that matter, or any topic for that matter, I always try to tell a story and I always try to construct a story. And what I mean by that is think about the different things that we've talked about this semester. Think about them in whatever context you have. If your background is in biology, try to take some of the content that we've discussed and apply it to that. So, one thing that is very important to me, and I would say kind of at the center of biochemistry, is amino acids, AA. Amino acids. And I always try to think about it as, okay, well, what are amino acids used for? Predominantly, we talked about amino acids as being the building blocks for proteins. Now, what do proteins do? Well, we kind of took two different directions with proteins. We talked about hemoglobin and myoglobin, HB and MB. Those are two examples of proteins that the most important part about them is that they are non-catalytic. So they don't catalyze any sort of reaction, but what do? They bind to something, move it, release it. Or they bind to something, hold on to it, and release it. They never change that thing that they bind to. So they just bind it, hold on to it, release it. Now, in addition to that, we talked about enzymes. Okay, well, what makes an enzyme different from something like hemoglobin or myoglobin? Well, one of the big things that makes them different is that enzymes are catalytic by nature, meaning that they will bind to something and then change it. They'll bind to this coffee cup and they'll open it. Um, just as an example. So, without going too much farther into that, I would say the a very good way to approach the material is try to organize the material and say, why did we learn this? Well, we learned this to build upon this. Why did we learn this? Well, we learned this to build upon this. So we learned about amino acid structures and amino acids because they give us a way to study proteins. Because if we understand individual amino acids, and are they polar? Are they non-polar? That is going to contribute to, well, what does that protein do? So we learned amino acids. We learned about amino acids because they build into proteins. And then we learned about enzymes as kind of a follow-up to that because then we were able to think about, well, enzyme mechanisms. And acid base catalysis, for example. Well, acid base catalysis, well, an acid donates a proton, a base accepts a proton. Well, then we can think back to our amino acids and say, all right, well, which amino acids are potentially involved in acid base catalysis? Are they going to be the polar amino acids or the non-polar amino acids? Well, let's think about the non-polar ones. Leucine or isoleucine. Those are hydrocarbons. Those aren't going to donate a proton or accept a proton. However, polar amino acids like aspartic acid, single letter code of D, or glutamic acid, single letter code of E, or what's the most popular amino acid when it came to acid-base catalysis that we saw, specifically with the RNase mechanism? Does anyone remember the amino acid? If you add the single letter, perfect. Joshua, or Jordan, sorry. Jordan, you're absolutely right. Histidine, H. Histidine is an ionizable R group, and whenever it doesn't have a proton, well, it's neutral, but it has a lone pair of electrons. So this is, I see you said got it, but this is the sort of thing that I highly recommend that you do. You know, kind of tell your own story from what we've looked at over the course of the semester. I hope that's helpful. And if you have any questions between now and the actual exam or after this class and the actual exam, let me know and I'm glad to try to meet with you. Okay, so we are going to jump back in to where we were. I'm going to toggle back and forth a little bit from this slide. So most questions would be from the practice exam. The practice exam that I'm going to post today will be a good way to think about. Um, I'm not pulling direct, or if I repeat a question from a practice exam on the actual exam, there's going to be a very small number of them. But practice exam, let's say here's our practice exam. If there is a question that is about calculate the pi of molecule, and I give you a peptide like AK, if you can calculate the pi of that, well, then on your actual exam, I'm going to label that AE, you're going to have a very similar question, but it's going to be giving you a different peptide, like ML, for example. So if you can do this, then you should be able to do this because that's a matter of looking at the um, sorry, that's a matter of looking at the pKa values, which I will provide to you. About how many questions will be on the final? Final will have 80 questions. Is it still only one note card? Yes. One 3x5 note card. So actually, you know what? This would be a good forum. If you want to take a minute and add to the chat something that you're thinking about that would be good to add to your note card, I'll give you a thumbs up or thumbs down. Like, yeah, that's a good idea, or maybe not. And you can do this directly or do a direct message or an indirect message. But things that I will provide you with is I will provide you with the pKa values of all your amino acids. The amino acid Krebs cycle, we're not really going to be asking questions about the Krebs cycle. The important takeaway from the Krebs cycle of the TCA, the tricarboxylic acid cycle, was what does it give you? It gives you NADH, FADH2, and some ATP. It gives you these electron carriers that you can use to get a lot of energy. So I would not waste space or not use space on my note card to like throw the Krebs cycle in there. I would include like a note, Krebs cycle, KC gives me, let's see that it would be 4 NADH plus 1 FADH2. I'd give myself numbers like that. What about amino acid structures of important ones? I want you to know all 20 of the proteinogenic amino acid structures. Now, with that in mind, what I would try to do, and this has kind of been an important or something that I've tried to say when we first talked about amino acids, is I'll say AA structures. I know that I mentioned like amino acid structures, and I have a tendency to refer to amino acid structures as kind of interesting or uninteresting. And most of your non-polar amino acids aren't particularly interesting to me. That's not to say that they're irrelevant, but if you think about glycine, alanine, leucine, isoleucine, those are all basically just hydrocarbons. And so they're not going to be involved in too many dramatic reactions. And so my point there is I would not try to commit those to memory. Whereas amino acid structures like aspartic acid or glutamic acid, the 1 letter and 3 letter codes of those, I would definitely make sure that I know those because those are good ones to be able to identify. In addition to that, these two structures are very similar to one another, much like leucine and isoleucine are very similar to one another. Um, so I would, when it comes to should I put amino acid structures on my note card, I would first do kind of a quick check. I don't know if anyone has like a Quizlet or something like that. I'd do a Quizlet and be like, which amino acid structures can I recall just by looking at them? And which do I have trouble with? Um, do I have trouble with lysine and arginine? Yeah. So what I'm going to do is I'm going to give myself a little bit of a note that helps me identify the difference between those. Because if you print up all 20 structures, that's going to occupy a good portion of your note card, and it just might not be worth your time. Glycolysis steps. I know the irreversible ones, but what about the other ones? Great question. When it comes to, oh, amino acid quiz on the Apple App Store helps with that. Okay, great, great. Um, Glycolysis steps. When it comes to the specific glycolysis steps, I would try to think about which ones first, yes, Riley, great point, are irreversible, but then I'd follow that up with which ones give me a product, give me something interesting. So, for example, let me go ahead. I'm going to give myself another slide, and I I don't want to rush through this conversation because I don't want anyone to be like, "Oh man, he answered other questions, but not mine." Um, I'll say glycolysis. Whenever I think about glycolysis, I think about the important points being one, irreversible steps, IRR. That's supposed to be IRR. Sorry for my speedy handwriting. So I think about my irreversible steps. HK for hexokinase, PFK1 for phosphofructokinase 1, and PK for pyruvate kinase. I would also think about, well, what are my products? And then I would clarify this. I would say, what are my products and my net products? So I'd ask those two questions of myself. Then third, I would ask, so I get NADH and ATP. How many of them? Then the third thing would be something like, well, where are there, you know, to me, basically the first four steps of glycolysis are the interesting ones, where you phosphorylate the sugar, you rearrange the carbon atoms, you phosphorylate the molecule again, and then you split it. After that, it's kind of just like general housekeeping. You have that dehydrogenase that's going to get you that NADH. So I would say RXN or reactions that make a product. That's kind of what I would prioritize. Okay. So those are some of the big points. Um, and I think that enzyme inhibition, oh, I love that. That would be great. So, you know, and make sure that you know like what whenever I'm drawing an inhibition plot, what I'm going to do is I'm going to kind of color code it for myself. And I'm going to say, okay, blue indicates no inhibitor. So that's just my reaction being catalyzed. I'd also want to go ahead and label my axes. So I'd say 1 over initial velocity and 1 over substrate. I'd also add a little detail and maybe put a circle or circle there and say this is 1 over V max. And maybe even give myself like a little point and say, okay, this is point 0.1, point 0.2. Just give myself some numbers to work with. So that would give me 0.2 equals 1 over V max. And give myself this math problem and say V max equals 1 over zero point 0.2, which is equal to 5. Okay, so that's one thing that's going to help me out. Next up is I'm going to go over here. Okay, and I'm going to say that this is, well, -1, -5. No, Joel, don't get fancy, Joel. I'm going to go with -2.5. There we go. That's nice. -5. And I'm going to say that point right there is equal to -1 over km. And of course, whenever you write this out, um, you're going to be able to make something that's a little bit more clean and something that you can follow. I'm going to say that is equal to -5. So whenever I do my algebra and rearrange it, I can say, well, -1 over km is equal to -5, or 1 over 5 is equal to km, which is equal to 0.2. Okay, so that's obviously that does not have an inhibitor present, but that's where I would, that's kind of my baseline. And if you're good with that, then you obviously don't have to add that. What about, okay, just a second, Melanie. I will get to that. Sorry for interrupting myself. Now I'm going to draw another line and say, okay, well, what type of inhibition? So this is plus inhibitor. And then I'll draw another line to illustrate another type of inhibitor. And I'm going to go with another color. Let's go with green. What if the inhibitor is here? Plus inhibitor. So that's going to give me a way to kind of jog my memory on, okay, well, if this type of inhibitor is used, what about if this type of inhibitor is used? What are the differences in these types of inhibition? How does my Vmax change? How does my Km change? Now, Melanie, what about the structures involved in muscle contraction? I think that it's a good idea to have those M lines and Z discs and understand like when the muscle contracts versus whenever it relaxes. But that's also something that I would not have as a super high priority. Because if you think about, the 80 questions that are going to be on this exam, you're looking at maybe 1 to 2 questions tops about that. Okay. Well, I'm sure that other questions will surface as we go through this. Um, what we're going to talk about today is, or whatever we cover today is going to be the end of the content for the semester. Um, I'm hoping that we can get to a little bit of biosignaling, but I'm not going to kind of rush through it. I just want to make sure that everyone's feeling good about beta oxidation because that's one of the places that we're going today. Now, what we've talked about most recently is lipids and fatty acids. Fatty acids being tremendous stores of energy. Lipids being molecules that, well, fatty acids are an example of them. Now, last time what we talked about in class was we talked about, well, how does fat get broken down? And we looked at this as kind of our general kind of flow of dietary fat entering the stomach. The liver produces bile acids and bile salts. Excuse me, but the gallbladder is where those bile acids slash bile salts are saved or stored. Whenever that dietary fat hits the stomach, it gets broken down with the help of these cholesterol-based, um, there we are, these cholesterol-based bile acids. Now, after that, we're going to have enzymes known as lipases that take those triacylglycerols that are your predominant components of dietary fats, and we're going to break those down into ionized fatty acids. So these things right here so that our body can actually process them. Those things, these R1 COO, R2 COO, those are going to be great sources of lots of energy. Now, if you remember last time, one of the things that we looked at was kind of the relationship between energy and carbons. For example, we talked about this in the context of like a nutrition, or sorry, a dietitian or nutrition. A dietitian is the real title. Nutritionist, I think, is the fake Instagram title. Um, 1 gram of carbohydrates are going to give you approximately 4 calories of energy. So it will give you that much energy. Whereas 1 gram of a fat will give you approximately 2 times as much. Um, and well, it's 2.5 approximately. But those fats are typically, or those fatty acids here are typically going to be 16 or 18 carbons long. Palmitic and stearic acid are examples of those. Now, lots of your dietary fat is going to come in the form of triacylglycerols. So you've got a structure like this right here that your body's going to break down. Now, lots of these triacylglycerols will kind of make this nice little sphere or spherical type of structure that looks like this right here, a micelle. Now, if you remember, the center of a micelle is very nonpolar because, well, it's just a bunch of alkyl chains, these hydrocarbon chains. And so these things cluster together really well because of the hydrophobic effect. Hydrophobic things love clustering with other hydrophobic things. Now, to disrupt this sort of molecule is a little bit tough. That's where the bile acids come in. They help break those down, and intestinal lipases are going to be responsible for basically removing individual components so that we can, well, process those triacylglycerols down into individual fatty acids. Now, okay, so where we kind of took off yesterday, and or one of the places that we left off was if you have a triacylglycerol, you have 4 components, 4 things. 3 of them are fatty acids. Those 3 fatty acids are going to be metabolized in basically the same way. The 4th thing is your glycerol, that glycerol backbone. That glycerol backbone is going to be broken down, and it's going to be broken down in order to make FADH2. That glycerol will also be broken down to form DHAP. That DHAP can be input into the inverse pathway of glycolysis known as gluconeogenesis. Okay, so that's what the fate of the glycerol from a triacylglycerol. In addition to that DHAP, we've got an electron carrier, this FADH2, which we can go ahead and extract more energy from that. Now, when it comes to the 3 fatty acids, those 3 basically identical fatty acids, what we need to do is we need to clip that long carbon molecule, that largely polar or non-polar molecule, onto something that the body can work with. And we use this enzyme, or we use this molecule known as acetyl coenzyme A, or coenzyme A, to basically flop the business end of that fatty acid onto. Okay, so we make this acetyl-CoA is what it's called. This group right here constitutes the acetyl component of that. So there's our acetyl-CoA, and that is kind of a general placeholder term for a fatty acid chain that we're going to break down. We're going to metabolize it to extract energy from it. Okay, so at this point, whenever we took that fatty acid chain and put it onto a coenzyme A to make our acetyl-CoA, which we see right here, we are still in the cytosol. Our process of extracting energy from a fatty acid is going to take place in the mitochondria, commonly known as in the mitochondrial matrix. So fat breakdown, think about that as taking place in the mitochondria. Now, in order to do that, we have to take this chain and get it into the mitochondria. We're talking about our inner mitochondrial membrane. Does anyone remember any of the molecules that can freely move across here? Freely move across that mitochondrial membrane? I'll give you a hint. There aren't many. Two of them are kind of low-hanging fruit slam dunks. Add it to the chat if you remember them, but I'm not going to wait for forever. Perfect. O2, CO2. Yeah, O2, H2O, and CO2 are your big three that can move across that membrane pretty easily. This membrane, this inner mitochondrial membrane, is super selective though. And so we can't take a largely non-polar molecule like a 16 carbon fatty acid and just say, go ahead, jump across the membrane. Instead, we have to basically affix it or attach it to something that can move across a membrane. And the thing that we attach it to is known as carnitine. Carnitine is an example of a non-proteinogenic amino acid. This is not involved in proteins or anything like that, but this is visualize or imagine this as just a molecule that you can easily modify and add a 16 carbon long chain to it. When you do that, you have this right here, your 16 or 18 or 22 carbon long molecule that you have attached to carnitine. The enzyme that attached that is known as carnitine palmitoyl transferase 1. CPT1 is a cytosolic protein. CPT2 is a mitochondrial protein. Now, anything that's attached to carnitine can be carried across the membrane because of the protein known as carnitine, or a, sorry, the carnitine carrier protein. This carnitine carrier protein will see anything that's bound to carnitine, move it across the membrane, move it across the membrane. It sees carnitine with 16 carbons attached to it, says, okay, great, I'll move you across the membrane because that's where you go. Whenever that is inside the mitochondria, well, then CPT2, essentially the same enzyme, but the mitochondrial enzyme, will release that 16 carbon long chain and attach it to coenzyme A, giving us this acetyl CoA inside the mitochondria. Could you give me a one in the chat if you feel good about the positioning of and your knowledge of the positioning of CPT1 and CPT2, where they are and everything like that? Okay. Now, one thing that over the years I've mentioned carnitine, and it sounds like such a familiar name. I want to stress though that this is not the same as creatine. If you're thinking, oh, well, I know creatine, creatine is an example of an amino acid. It is a non-proteinogenic amino acid, but creatine is not involved in any sort of fat breakdown. Carnitine, however, is. They look similar in name, but they are different in structure and different in function. People take creatine as a dietary supplement. People also take carnitine as a dietary supplement. The efficacy, I love that word, efficacy of carnitine, like orally ingested carnitine, I really don't know anything about that. Creatine, on the other hand, that one's pretty well studied, and I think that that does have positive benefits. Okay, so when CPT2 acts on your carnitine with that fatty acid chain, you've effectively got your fatty acid chain into the mitochondria so that it can be utilized. Now, that's where beta oxidation comes in. Okay, so I want to introduce you to the pathway of beta oxidation. I wrote on this slide, but I'm going to go to another one where we have basically the same information, but it's a little bit cleaner. Okay, so beta oxidation, the things that I want you to know. The first thing is that it takes place in the mitochondria. Beta oxidation, nice way to think about it is FA or fatty acid breakdown. Okay. Now, when you think about beta oxidation, I want you to think about this also as a high energy yielding process. Now, we have a fatty acid that is covalently bound to coenzyme A. This is where that FA-CoA comes in. Now, if we jump back a couple of slides, it is very common to look at this whole thing right here and simply identify that as CoA. So that whole structure, we don't have to know that structure. We don't have to memorize that structure or anything like that. You don't even have to visualize it, but just know that that is CoA. So whenever I talk about CoA, that's what I'm talking about. Because when it comes to fat breakdown, that's all that it is. CoA. We will not really use any other part of it. Now, what we care about when it comes to fatty acid breakdown and beta oxidation is really the CoA, or sorry, the Fa. So here is my Fa. Now, just to give you a little bit of more context, this is, I'm drawing 1, 2, 3, 4, 5, or 1, 2, 3, 4, 5. So that's 5, oh, dang it. I didn't give myself enough space, so I'm going to draw below. Oxygen, oxygen. Okay, so there's my fatty acid. This oxygen right here, I've replaced with this S-CoA. Okay, so that's where that is coming into play. Now, whenever it comes to Greek letters or nomenclature, um, we referred to our proteinogenic amino acids with a Greek letter also, or they are also identified as this amino acid. Does anyone remember what this amino acid is? Whenever I say using a Greek letter, the proteinogenic amino acids that we talked about, we also referred to them as alpha amino acids. Why did we call them alpha amino acids? Well, the reason that we called them alpha amino acids is because the R group was coming off of that alpha carbon. The alpha carbon is going to be the carbon that is adjacent to a carbonyl group. So here, I'm going to do some erasing. I'm going to scribble out the S-CoA because we're going to kind of ignore that for the time being. Here's my carbonyl carbon. Here is my alpha carbon. Carbonyl carbon, alpha carbon. Give me a 7 in the chat if you follow that. You're good with that. Okay, great. Now, it's important to see that alpha, the beta, and the gamma carbons in a fatty acid. Now, this right here is an example of a fatty acid that, well, N length of it. It could be 16, it could be 18, it could be 12 carbons long. Beta oxidation is going to be where we basically clip off, and this was always helpful for me, was in every round of beta oxidation, we are clipping off 2 carbons. So the way that I always kind of think about that is we are taking the carbonyl carbon and the alpha carbon, and we're going to go through to release those. That's kind of the objective. That means that this beta carbon, after one round of beta oxidation, that is going to be my new carbonyl group. New carbonyl carbon. Okay, so that's kind of your primer. Now let's go through and look at these different steps where this is taking place. So in the very first step right here, what we are going to do, and I'll revisit some of these things, and if anyone has any questions about these, I will be happy to talk about it again. I'm going to draw that new carbonyl carbon after R1, round 1. Okay, so the first thing that I'm going to do is I am going to use a dehydrogenase. Could anyone tell me what type of reaction a dehydrogenase enzyme is going to catalyze? Is it a transferase? Is it a hydrolase? No. Dehydrogenase. I'll give you a hint. It starts with an R. And notice how we use FAD and FADH2 in the process. FAD and FADH2, reduction. Yeah, this is a redox reaction. FAD, anytime you see FADH2 and FAD, I want you to think, this is a redox reaction because FAD, FADH2, electron carriers. Okay. So this is a redox reaction. Now, looking at what we have, or looking at what's changing from this molecule to this molecule, is we're introducing a double bond between our alpha and beta carbons. So we're introducing a double bond, and in the process, we're transferring electrons from this molecule onto FADH2. Okay. I said previously that FADH2, anytime you get that, boom, you've got something that you can later on use to generate some energy and ultimately make ATP. Now, I'm going to have you think about the first reaction as, okay, we generated FADH2 from that first step of beta oxidation. Now, what I want to do after that is move on and say, okay, well, what's the next step? And keep in mind that our beta carbon is going to become a new carbonyl carbon. So if something is going to become a carbonyl carbon, we need to get an oxygen on there. Give me a one if that makes sense. You've got to get an oxygen onto something if you want it to be a carbonyl carbon. Okay. How are we going to do that? Well, what we're going to use is water. We've got this carbon number 2, carbon number 3, where we have a double bond, carbon 2, carbon 3. Oh, we're going to throw some water on there. We're going to add this hydroxide group to carbon number 3 and the other hydrogen. So if you think about H2O as being H and OH, well, the OH goes on to carbon number 3, which is our beta carbon. And the H is going to go on to carbon number 2, which is our alpha carbon. Okay, so hydratase is going to be the second enzyme in the rounds of beta oxidation that's doing this. Now, could you give me a 9 in the chat if you're following this alpha, beta, 2, 3 designation? And any other character if you're mixed up on any of them. Okay. So now that I have this molecule, which I've just added an OH to, I'm going to use NAD and I'm going to generate NADH. This is a DH, a dehydrogenase. Now I'm going to go back to give you the full name of it, but I'm not expecting you to know the full name of that. The full name of that enzyme is 3L hydroxyacyl CoA dehydrogenase. Okay. What type of reaction is this enzyme catalyzing? That I just put a star by this DH. Add it to the chat. It's another redox reaction. Oh man, these oxidation and reduction reactions, well, we know that oxidation and reduction reactions can generate a lot of energy. We've got another electron carrier. We've got NADH, which we can subsequently or later on use to generate some ATP. Okay, so in these first 3 steps, we've done a redox reaction where we can insert a double bond. Then we disrupt that double bond by adding water across that double bond. The hydratase does that. Then we've got this dehydrogenase where we generate some NADH. Now, effectively, that gives us 1 carbonyl group, 2 carbonyl groups. I told you previously that carbon number 3 here is going to be our new carbonyl group. Do we have a carbonyl oxygen on that carbon number 3? We do. So that kind of fulfills what I was kind of getting at. Now, the last thing that we need to do is we need to use an enzyme known as beta ketoacyl CoA. This enzyme is going to bring in coenzyme A. And what it's going to do is link up or form a covalent bond between coenzyme A and what is carbon number three. And whenever that happens, that is going to displace the two carbons and coenzyme A. Remember what I said about every round of beta oxidation, you're cutting two carbons off at a time. Here is where we do it. Beta ketoacyl CoA displaces this first carbonyl and carbon number two. So carbons number one and two from that original chain. So essentially what that means is if we started with, I'm up at the top now. If we started with a fatty acid that was 16 carbons long, tell me in the chat how long is this fatty acid chain that I just circled in red? We started with something that was 16 carbons long. Remember, this N is just indicating that it goes on in perpetuity. It goes on for forever, or it goes on as far as we want. We started with something that was 16 carbons long. How many carbons did we remove from that 16 carbon chain? In every round of beta oxidation, we remove two carbons. So if we had something, if we had a fatty acid that was 16 carbons long, we removed two, we're looking at a fatty acid that is now 14 carbons long. Could you give me a 6 in the chat if that makes sense, or a W in the chat if I'm confused? Okay. So it looks like people are clear on that. Now, in the process, I want you to think about beta oxidation, and I want you to think about products. What are the products that we got from one round of beta oxidation? Well, the direct products that we got FADH2. I said that later on we could use that for ATP, but in terms of just the round of beta oxidation, we got FADH2 and NADH. And this thing right here. What is that? That is known as acetyl CoA. Two carbons, one of them being a carbonyl carbon and the other being a methyl group. That's acetyl CoA. Could you give me a three in the chat if you agree on these three products from one round of beta oxidation? Oh, and then the other thing is, well, you have a fatty acid that's slightly shorter. Okay. So acetyl CoA, though, have we seen acetyl CoA before? Spoiler alert, we have. Where did we see it? We kind of went through it a little bit briefly, but look at this right here. Melanie, you're absolutely right. The Krebs cycle. So acetyl CoA. Oh man. Let's see. One carbonyl and one methyl group, and then S-CoA. Is that the exact same? Yeah. Could anyone tell me where the Krebs cycle takes place within a cell? Basically, I'm just looking for, is it the cytosol or the matrix? I'll give you a little bit more direction here. If we look at this structure, it's in the matrix. Okay. So let me get this straight. From from one round of beta oxidation, we directly produce one FADH2, one NADH. Okay, cool. From an acetyl-CoA, well, we're going to be able to plug that into the citric acid cycle, and we're going to get four NADH, one FADH2, and one ATP. It's truly GTP, but it's effectively the same. So when it comes to breaking down a fatty acid, you get all of this. I'm going to go ahead and scribble that out. You effectively get all of this from one round of beta oxidation because the products of that one round of beta oxidation can go into the Krebs cycle where you can generate more and more NADH. Could you give me a I in the chat or if you follow this? And a Z in the chat if you're confused about maybe where something fits in. Okay. Now, I'm not worried about this right here. Instead, what I want you guys to focus in on is the overall math. And that's what's on this next slide. So we have palmitic acid. That is the common name for a 16 carbon long saturated fatty acid. Now, a fair question for you to ask is, well, saturated fatty acids. Are saturated fatty acids metabolized differently from unsaturated fatty acids? Yes, they are. That's beyond the scope of this class. If you go on to take metabolism, you will get into metabolism of trans fats, metabolism of unsaturated fatty acids as well. Now, for the beta oxidation or the breakdown of a saturated fatty acid, let's look at this. How many rounds of beta oxidation will you do? I touched on this the other day and I want to bring it back. If you have a 16 carbon long fatty acid, your first round will give you a 14 carbon and an acetyl-CoA molecule. So that's 2 carbons. Your second round will give you 12 carbons and an acetyl-CoA. Third round, 10 carbons and acetyl-CoA. Fourth round, 8 carbons, an 8 carbon long fatty acid and an acetyl-CoA. Your fifth round, 6 and an acetyl-CoA. Fourth will be a 4 carbon and a fatty and an acetyl-CoA. And your last round of beta oxidation, you're starting out with a 4 carbon long fatty acid or fatty acyl-CoA, and you will split that into a pair of acetyl-CoA molecules. Could everyone give me a 9 in the chat if you understand that 4 carbons will be split into 2 identical molecules? Now, that's an important point to remember because we're not taking, like, as soon as you break it down to 2 carbons, you're not going to go through any more rounds of beta oxidation. So that's why we end up a 4 carbon long molecule gets split into two acetyl-CoA molecules. Okay, so to completely metabolize 16 carbon palmitic acid, you'll go through 7 rounds of beta oxidation. And from those 7 rounds of beta oxidation, you will get, and this nominal right here is basically approximately the approximate outcome. You'll get 7 FADH2. Now, those 7 FADH2 that you generate, those will get you 14 ATP. Now, this 7 right here is from or directly from the beta oxidation, not from the fates of anything like that. Okay, so that FADH2 will get you 14 ATP. You'll also get 7 NADH. Those 7 NADH will be used later on to make 21 ATP. Now, the real, could anyone tell me what is the real money maker when it comes to beta oxidation? What is the thing that's going to get you the most energy from these three right here? Is it FADH2, NADH? No, it's not FADH2. FADH2 will get you basically one FADH2 molecule is going to get you 2 ATP. One NADH molecule will get you 3 ATP. What will acetyl-CoA get you? One acetyl-CoA molecule will get you 4 NADH, one FADH2, and 1 ATP. Melanie, you are jumping ahead, but yeah, you're pretty darn close there. Yeah, great. So acetyl-CoA, that's the real moneymaker from our 7 rounds of beta oxidation. We will get 8 acetyl-CoA. Now, this is a number that always kind of distracts people because they're like, wait, why is it not 7? Well, the point is the last round of beta oxidation gives you 2 acetyl-CoAs. From those 8 acetyl-CoA, you'll get, when you plug these into the Krebs cycle, you'll get 8 GTP. Those GTP are basically the same as ATP. That's why I've referred to it as ATP from it. Oh, Joel, stop. Sorry. You'll get 24 NADH. Oh, I'm sorry. Yeah. Okay. From those 24 NADH, you'll get 72 ATP. Those 8 acetyl-CoA will get you 8 FADH2, which effectively will be 16 ATP. So I need to go back and make one change for my numbers here. This shouldn't be a 4. I apologize for that. I'm going to change that. That should be 3 NADH. So from one acetyl-CoA, you'll get 3 NADH, 1 FADH2, and 1 ATP. Now, that's all going to add up to, from one 16 carbon long fatty acid, you're going to get approximately 131 ATP. So this is where it comes to, well, what's better, to consume fats for energy or sugars? Well, fats are better from the standpoint of you get a lot of energy from them. And this is basically how that, through that process known as beta oxidation. Now, what we need to do is we need to break it all the way down to CO2, and that's what the Krebs cycle helps us with. Now, where this NADH that we use, or where this NADH is going to go, is into this right here. This is known as the electron transport chain. The electron transport chain is just that. It's a system that transports electrons. When you transport electrons, when you take electrons from one thing to another thing, well, that thing that releases those electrons gets oxidized. The thing that gains electrons gets reduced. There's lots of energy transfer there. I always think about metals like sodium or lithium. If you put those in water, they're going to get oxidized, and when they do, they're going to release a lot of energy. Potassium, sodium, lithium, if you throw them in water, they're going to basically start on fire because that oxidation reaction is releasing so much energy. We've got that same thing, but this is the substance, this NADH2, that is going to lead to the release of a lot of energy. So we're going to move electrons from NADH all the way through these different protein complexes. These protein complexes, in addition to moving electrons, are going to move protons from the mitochondrial matrix, where our Krebs cycle and beta oxidation have taken place, into the intermembrane space. So that's the space between our two mitochondrial membranes. Now, this number here is important. How many total protons are moved across the membrane with the help of NADH? 10. Okay, so we got 10 protons. It might not seem like a lot, but Here is this protein complex known as ATP synthase. What ATP synthase does is it is driven with the help of proton movement. So at the bottom of this slide, in this image right here, we're talking about the intermembrane space. I'm going to label this as IM space, the intermembrane space, where we have just moved a bunch of protons. Now, protons will funnel from the intermembrane space into a spot within this protein. Those protons will subsequently be moved to the other side into the mitochondrial matrix. And in the process, this protein known as ATP synthase makes these minor movements. A total of 9 protons will be moved from the intermembrane space to the mitochondrial matrix. And when those 9 protons are moved across, we're going to generate ATP, a total of 3 ATP. So essentially, 9 protons moves, or sorry, 9 protons moving across the membrane is going to produce 3 ATP. Could somebody tell me in the chat how many protons will be moved into the intermembrane space with 1 NADH? So the answer for that on the previous slide, 1 NADH molecule. Here's our NADH. And just visualize this as, well, this NADH is going to move electrons from itself to something else, from itself, or from that to something else, from that to something else. And we're going to go through this. Okay, so these red arrows that I'm drawing in is just to show the movement of electrons. Is everyone clear on that? Give me a ZZZ RQ if you follow that. Okay, okay. So NADH has moved electrons. Now these protein complexes. Oh, Emily added a ZZZR. And also with a ZZRQ. Was asking for a ZZZRQ. Okay, I'm just joking. Okay, so these protein complexes, think about it as it's doing 2 things simultaneously. It's like, okay, cool. I can take these electrons and move around to these different complexes. And what that is going to do is that's going to give me the energy to move protons out of the matrix into the intermembrane space. So NADH, when it initiates this movement of electrons, when NADH gets oxidized, electrons start moving. Okay, and the flow of those electrons through these different protein complexes leads to the movement of a total of 4, 8, 10 protons. Could you give me a QQZR in the chat if you follow that? Okay, okay. So essentially, you could think about that as 1 NADH moves enough protons. And keep in mind, what is NADH doing? NADH, all that it's doing is it's getting oxidized. So it's releasing electrons. When it releases those electrons, the protein complexes here say, okay, cool, you've released those electrons. Now what I'm going to do is move protons across. Okay? These protein complexes will move a total of 10 protons. Those 10 protons that are pumped here, where I put a big X, those 10 protons will drive this protein complex, this ATP synthase, and drive it to move, undergo these really interesting and complex movements that lead to the production of ATP in the mitochondria. So my question, one of the questions that I like to ask is, where does ATP synthase produce ATP? And you got basically where ATP, I'll say made. That's really not a very good, well-worded question. Where ATP made? You've got options. Matrix, IM space, then I don't know. That's an option. And then last but not least, membrane. We've got those four options: the matrix, the inner membrane space, the I don't know, or the membrane. Okay, think about those. I don't know is out. That's not an option. So where is the ATP being produced? Is it in the membrane? No, that's not an option because in the membrane, that's where the protein is embedded. Melanie, you are absolutely right. The matrix. The ATP is actually generated in the matrix. And after all, what do we call the mitochondria? Add that to the chat. What do we call the mitochondria? Powerhouse of the cell. Powerhouse. Powerhouse of the cell. Somebody could have just said mitochondria and they would have trolled me well. I appreciate that. But yes, the powerhouse of the cell. So that's where all that ATP is being generated. Now, a pivot on this question, rather than where ATP made, I could say where protons pumped. Pumped. Come on, Joel. Where are protons pumped? Is it the matrix? No. Is it the, I don't know? No. Is it the membrane? No. The protons are pumped into the intermembrane space by those different protein complexes. Now, a visual of, so what I want you guys to take away from all of this is I want you to be able to draw a connection between the breakdown of a fatty acid. What does it get you? What are the important parts of a fatty acid? Well, FADH2, NADH, and acetyl-CoA. So overall, if I said you have a 64 carbon long fatty acid, what are you going to make from all of that? Well, my first question is, how many rounds of beta oxidation are you going to do? How many rounds of beta oxidation? Okay, so a 16 carbon long fatty acid gets 7 rounds of beta oxidation. Okay, so the way that I always kind of think about that number is I divide it by 2, subtract 1. Patricia says 31 rounds. You're absolutely right. You're going to do 31 rounds because 31 times 2 is 62, plus 2 will get us to 64. So 31 rounds. Now, what are we going to generate from that? We're going to generate 31 FADH2. We're going to generate 31 NADH. And we're going to generate 32 acetyl-CoA. That one, or FADH2, so from a 64 carbon long fatty acid, well, you get 31 FADH2. So that's going to be approximately 62 ATP. From the NADH, all the NADH that you're going to make, all 31 of those, that's going to be about 93 ATP. From the acetyl-CoA, well, you can kind of extrapolate and expand that out. That's going to be, let's see, 3 NADH, 1 FADH2, 1, I'm going to say GTP slash ATP, but there's 32 of that. So that means I'm going to get 96 NADH because 32 times 3 is 96. Then 32 FADH2, and then 32 ATP GTP. Now, these molecules, this NADH and FADH2, that's a lot, but what matters is these oxidation reduction reactions. Each NADH is going to release electrons, and the release of those electrons is going to cause the driving of movement of protons from the matrix into the inner membrane space. Now, for every 9 protons that you move, you're going to be able to generate 3 ATP. All of these structures, these last few slides, are all about what that protein structure looks like. Now, this is the part of the protein that is in the mitochondrial matrix. You have 3 different spots on this protein. You've got a loosely binding site, an open binding site, and a tightly binding site. And then you have this little, it kind of looks like, really, it looks like a nose or a kind of like a Hershey kiss or something like that, that this structure right in the middle is going to rotate. And that structure rotating is going to cause a change in the conformation of these different binding sites. And that's going to cause ADP and inorganic phosphate, this is within the mitochondrial matrix, to make ATP. So the way that I always kind of think about this is this loose binding site. Could anyone tell me what it is that you use to make diamond from? Or what can you make a diamond from? Carbon, yeah, yeah. I'm thinking about like coal. If you put coal under enough pressure, it turns into a diamond. Okay, so imagine that we've dumped a bunch of coal into here. This protein that's in the middle is going to undergo a change. And that is going to cause this ADP and PI to be forced together. It's almost like these lumps of coal that you're forcing together under enough pressure that then pops, and then you got a diamond out of it. The diamond in this case would be the ATP. Does that analogy work for anyone? Give me a ZBV, if that makes sense. And if it doesn't, don't put Z. Okay, ZBV. Okay. Yeah, ZBV is what I was looking for. It doesn't make sense. That's fine. Okay, so that's where I'm going to go ahead and stop the material for this semester. That's where I'm going to stop the lecture. So what that means is that your final exam is not going to have anything related to biosignaling. I'm not going to spend the last 12 minutes talking about biosignaling and try to make that work. I'm going to stop my share and I'm going to let you know that I'll have the practice exam and kind of a study guide up by about 5 p.m. today. That's my goal. That's my plan. I should be ready for that. I should be able to get that. Does anyone have any questions? And if you don't have questions now and you want to meet some other time, I'm up for that. Just let me know. I will be posting this recording on our Canvas site later today. The project is indeed optional.